Hi everyone. Sorry, I just updated my slide. So we need to wait for a bit.
I'm waiting for connection.
Yeah. So while waiting, just to introduce myself, I'm Sudat from the National University of Malaysia. This is the work that basically done my first PhD student. I'm so proud of her. She's Fatima, but she's can't be here. Uh, why don't we work on rice because this is something that we actually are focusing on. And we go for the next slide.
I think so. No?
Yeah. So we focus on rice because so just next is it?
Just this. Oh, sorry. Yes.
Yes.
Yes. Here. Here. Here.
Okay.
First.
Yep. So we focus on rice because rice is a staple food. Almost all of us have it. I'm just showing some of the pictures from Malaysia food. You have coconut rice. It's super nice. The other one is Hainan chicken rice that we live in. Apart from Malaysia, we have other cuisine that features rice. One thing to mention why work on GWAS is because of SSL. Basically, in Malaysia, we can't really meet the demand of SSL. Basically, we don't produce enough of rice, and all of this is because of the flooding and also the drought you have. So apart from that, you can also get information, the stress from the salt and even from the biotic ones, so from the fungus, from bacteria, etc. So all of this is very interesting, but however, if you actually want to understand the stress adaptive mechanism, usually we will sequence the genome, and then you have the genome will conduct something called genome-wide association studies, and then after that, this can pinpoint the candidate genes and mutation for you. So usually when you do this kind of analysis, it will give you a lot of mutations, a lot of genes. You're not sure what to do. So I come up with an idea, is it possible for us to map all of these mutations on protein structures? And by doing so, you can prioritize some of the functional important ones. However, before this was actually very hard. So for rice, previously it's only 100 protein structures. So that said, but recently you have AlphaFold and also the TED domain also annotations. You can help a lot. So thanks to John Jumper and Demi Hassabis, they developed AlphaFold, and then Samir Valenka helps, and they have this AlphaFold protein structure database. And after that, Christine Orango and David Jones, they started working on TED. Basically, what TED do is they have three different algorithms. All of these different algorithms, they will figure out what's the best domain chopping from them. And then after that, there's an agreement between two of the software. They will just get the assignment from there. And this is called the Encyclopedia of Domain. So for those of you that still confused what I'm saying, you have one protein structures, you chop them into domains. And then after that, you need to check your domain, make sure they have of high quality, check the sangradine structures, packing, copularity, and after that, you have the good quality one. So in the studies, we want to understand what's the impact of mutation on stress-associated mutation. So I told you there's only 100 odds of rice structures. If you look at domain, it's 360. With TED, you can now improve it 276 fold. And in the next, we can do the next part. The next part, we actually use GWAS mutations. We got it from GWAS Atlas, thanks to the China National Center of Bioinformation. And then after that, we narrow down and take all of the plant associated one, and then we are taking the stress category. And then after that, we map them to domain region, and we found there's only like 150 odds of them that actually map to structural domain. Probably some of you will be thinking why a lot of them are not inside the data. The second one is quite obvious. Maybe the models of them are not quality. The other thing is because the other reason is basically they are outside of the structural regions. So there's some studies that have found most of the rice, they have mutations near the non-coding regions, near the enhancer, the are part of the regions that's non-coding. And this is our data, 100 odds. So about 130s are biotic stress, and then the others are abiotics. So the highest one is actually from bacteria blight, and then after that we also have plant hopper and also sheath blight, that's a fungus and also virus. We run a penta analysis using a Paul Thomas method, and then we found out there's like enriched in antimicrobial resistant proteins. This is not surprisingly because we have a lot of biotic stress kind of data. We also found a lot of transmembrane signal peptide. They're usually near the surface, kind of normal to have them. You also have a lot of enzymes. And then after that, we use MAPRED2 basically to figure out this mutation are actually deleterious or not. Some of you will be thinking, why most of them are actually non-pathogenic? They're not deleterious. Initially we were shocked, but after thinking about it, most of these mutations should be gain of function mutation, and because we won't want the mutations to change the function of the protein drastically, so it's just tuning the effect a bit more. So that's more like gain of function mutation. And then after that, we also ask whether these mutations are exposed or not. So mutations that are exposed, they usually lie close to the functional site, and that will be the places like protein presentation site, ligand binding site, etc. We also remove some of mutations that are not really having a good Grantham score. We use Cecil-Grantham method. Grantham, so anything that's less than 50, you have similar for your physical properties. And then we are left just with 95 mutation. For all of these mutation, we want to understand whether they are close to a functional site or not. And sadly, in plant, we don't have a lot of known functional site data, so we need to predict them. To predict them, we use all of the known software. Probably one of the developers here. We increase the confidence level for a lot of algorithm because we want it to be more stringent. And you have all of this side. And then after that, we figure out these mutations are actually close to our functional site or not. So we found out of the 75 mutations, 3 of them are near PTM, 9 allosteric site. 45 of them, so this is the highest one, near protein-protein interface, ligand binding site. And some of them are close to FUN site. This is basically evolutionary conserved position where it's been shown that it's been highly enriched in protein-protein interactions, ligand binding site, catalytic site. And we found 70 mutations that are unique and they are close to functional site. Some of you may be questioning, are there any overlap between them? We found about 21 of them that actually overlap with more than one predicted functional site. This is one example you have. This is a transferase. We got a good model. It's related to bacterial blight. We run Serrano's lab FOVAX, and then after that, we figure out this mutation is stabilizing. We also found a predicted allosteric site near there on top. Transferase has been shown to be implicated in the stress modulating, maintaining the redox balance. And next thing, probably a lot of you guys will be wondering, and probably you will do that the same. So I focus on the protein perinaria site. And in order to do that, we identify the binary protein interactions using string, and then after that, we model them using AlphaFold. And we found 12 models to be highly quality, and we also checked their Portage score and PISA score. And we use Douglas Spears and David Asher's method, MCS and PPI2, to figure out these mutations, what's the impact on the complex binding. And we found 6 mutations to be highly, increase the complex binding. So this is one example that have peroxidase and CAD. So this has been implicated in the rice blight disease. This is something called arsolani. If you're actually looking at it, it's causing this kind of lesion. With this lesion, the plant will die. And this peroxidase and CAD basically is part of the building of this lignin. CAD basically provides the substrate, and peroxidase is the one that produced the lignin. So some of you, this is the thing, cell wall lignin. Yep. And this is a mechanism, CAD and POD is at the bottom. So this is the model that we gathered from AlphaFold. We checked the score. We also found one of mutation A116 to be affinity enhancing. We do structure analysis. We found more and studied the hydrogen bond. We found like the Newton actually have more hydrogen bonds and a new hydrogen bonds forming and more interactions happening there. Probably some of you will be thinking, why suddenly two enzymes actually want to increase the binding of them? And we can back and think about, is it possible something called substrate channeling? So what happened here is in some kind of like biologies, you have metabolon forming. What happens here is two enzymes actually come together, and when they're together, it's easier for them to flow the metabolite and all the thing they have produced from one enzyme to another. So we're seeing this kind of pattern when increased binding. So this is interesting, but I think we need more like experimental validation on this topic. Conclusion, AlphaFold did help to increase the number of structural domain. We examined 140 and we found about 70 of them that close to mutation, close to functional site. And these will be very helpful. And this is something that's probably a lot of you will be, will be want to know, because all of them will be helpful if you actually want to go for gene editing, marker access kind of breeding, stuff like that. And we're now looking for collaborator to validate our mutations. And just 2 days ago, this is just a promotion, not this talk. We have trying to write. We wrote a review paper on plant, looking at the stress adaptations kind of mechanism. It was a very, very hard paper to write because there's not really a lot of plant structures that's available. So if any one of you want to start working on plant, it's a good time to go into that. This is the student I'm so proud about, Fatima Shahid. And my collaborators is Neil Strong from ISMB, Nicola Bordin, Christine Orango. My other collaborators, not related to this project, Mike Brown. Yeah, thank you so much.
Thank you. Questions? I have a quick one. You're extracting your data mutations from the GWAS catalog. The mutations of areas that are in GWAS catalog that you extracted, are they always the actual mutation, or could they also, at least some of them, be just some tag SNPs?
Yeah, it can be some of it as well. But we try to go for the plant ontology and the thingy where it's plant stress. But maybe they may link to other things as well. Like, more validation needed. That's why. Yeah.
Other questions? Okay, if not, thank you.
Thank you so much.